Okay, briefly we'll talk about genetics here. The body, its tissues, its functions, everything is under the control of the genetic processes, specifically DNA, which is made up those four bases, the A, the T, the C, the G that you talked about, and the combination of the DNA tells the body what to do. Well, genes are pieces of the DNA that have the coded instructions. You know, one AT pair doesn't tell the body to do anything, but a grouping of these DNA bases is a gene, and that has instructions on it. They need to get together to make a small piece of DNA to have those instructions. Now, when we put all the genes together, we come up with chromosomes. Humans have 23 pairs, or 46 chromosomes. One half comes from the biological mother, and the other half comes from the father. Cells divide by mitosis and meiosis. Mitosis is the division of the cells that result in identical cells. You know, you start with one cell, it breaks into two identical cells. These are the body cells, or the autosomal genes, chromosomes. And then there's meiosis, which will result in cells that only have half the needed genetic information. These are the gametes, the sperms and the eggs. Okay, as we said, genes come in pairs, one from each biological parent. Then the individual has a genotype, which is the actual genetic makeup, and they have a phenotype. That's how the genes are expressed within an individual. Here's an example. Okay, a person carries two genes for everything, including their blood type. A and B are the dominant blood types. O is recessive. So, a person may get an A from their mother and an O from their father. That makes their genotype AO. But their phenotype is A. The expressed blood type, what we call the blood type, is type A. That's a phenotype. So we have dominant and recessive genes. If a person carries a dominant gene, that's the gene that's going to be expressed. In the previous example, A is dominant. So A is the blood type. Recessive genes are only expressed if both genes within the pair carry it. So, for a person to have a phenotype of O, both genes within their blood would have to be type O. Alright, here's another example. Eye color. Brown is the dominant eye color. A person with one gene for brown eyes and one for blue eyes is going to have brown eyes. Their phenotype is going to be brown eyes. Only people with both genes for blue eyes are going to have a blue eye phenotype. Alright, let's look at the Punnett square. That's the way that describes the possible gene combinations that can be inherited by any given biological mother and father. Here's the one for eye color. In this case, the mother carries two genes for blue eyes. The father carries one gene for brown eyes and one for blue eyes. When you look at all the possible combinations, you can see half of them have two blue eye genes and half of them have one brown eye, one blue eye gene. These are the genotypes for their possible children. So what are the phenotypes here? Well, we see two of them have brown eye phenotypes, and two of the possible children would have blue eye phenotypes. Chromosomes can be damaged. Now, copies are made of these damaged chromosomes. They're going to be replicated, and each one of the subsequent daughter cells, chromosomes, are going to be damaged. because they are identical to the original damaged one. Pieces can be deleted or removed or moved to a different spot, or a piece can be duplicated in the wrong place. Okay, here are two problems that sometimes occur with duplication. The first one is mosaicism. It's the presence of two or more different lines or sets of chromosomes within one person. This happens early in fetal and embryonic development. One of the chromosome pairs mutated, and this is early in development. So that line keeps replicating that mutated set of chromosomes. So that person is going to have two different lines of chromosomes, one of them being apparently normal, the other one is abnormal. Well, an example of this would be, some people have mosaicism with Down syndrome. Not all of the cells carry the trisomy 21 abnormal chromosome. Some of them may be normal, not have that trisomy 21. The others have the trisomy 21. Well, it depends when in the embryonic life of this individual did this mosaicism occur, and where did it occur? Were they the genes that eventually made the brain? Then this individual is going to have some cognitive delay. Is this something that had to do with the development of the way their face looked? Well, then they may have the typical face that is common with people with Down syndrome. But other parts of the body might not be affected. And the second problem that we're going to discuss here is aneuploidy. I probably said that wrong, but there you go. It's the wrong number of chromosomes. For instance, Turner syndrome, rather than having an XX or an XY, there's just an X. That other one has been lost, has been deleted somewhere early in development. And the second one that we just talked about is trisomy, having 3 genes instead of 2 at one place. Down syndrome has an extra 21, number 21 chromosome.